And welcome to day 23 of MetPhys 2020, Metabolic Physiology in Isolation, where today we're going to hear from Dirk Brenner about his fascinating work in Tregs. I've said this every day, but in case this is the first time you're watching this series or recording of this series, metabolic physiology in isolation has become a new model for truly inclusive global science. We've been able to provide excellent content in a very cost-effective manner. The cost is completely taken care of by the White McGarry Lab, and we're proud to do that, so that anyone, anywhere, at any time can watch great science talks and be inspired and generate new ideas that may solve some of today's most pressing problems, from cancer to diabetes to fatty liver disease. And as we heard from Peter Walter earlier in the week, even things like traumatic brain injury may have their roots in mechanisms of metabolic physiology. It's also green. We're not incurring a impact on the earth by traveling to a conference. And so we're not trying to change the fact that going to conferences is important and valuable, but we're adding a new model so that we can also include those people who may not be able to attend. I apologize, yesterday there was a major Zoom issue. The Zoom issue was caused by an issue with the switch of the subscription to Zoom that the White McGarry Lab is taking care of. When they transferred us for this month, they didn't add the webinar feature, so we couldn't run through Zoom yesterday. Fortunately, karma was abound, and because we had pre-recorded that talk with Miriam, we were able to immediately upload it to YouTube for you all to watch and not be too late with that. But we're back to Zoom now. After some angry phone calls, we're back on track. And so I apologize to Cassidy, who has been working very hard to get her Inception prize. I'm going to give her a mulligan for missing yesterday. It was absolutely my fault and not hers. But here is the photo she shared the day before. And what I really like is that even her Barbies are practicing good preventative measures for COVID-19. Look at that face mask. We also saw a new entrant into the best Zoom background from Alex Muir on his talk earlier in the week. I'm going to be putting up a poll at the end of the week and seeing if anyone else thinks that that is better than Isaac Harris, who is in the lead at the moment for the best background. And so we'll see how that goes. Stay tuned for that. More to come. Every day when we have these talks, and even if you're watching the recording, the way you can ask a question, and you can ask this question even if you watch this talk in 2021 or 2023, is you go onto Twitter, if Twitter still is around in those days, in that time, use the hashtag MetPhys2020, and if you know the speaker's handle, you include them, at the very least include myself, and your question or your comment. You can ask as many questions as you like, make as many comments as you like. In fact, the comments don't even have to be related to the work. And if you just want to show us a picture of your cat or your dog, we enjoy those as well. They're helping everyone to kind of get through this isolation moment. I've shared before that we're also doing MetPhys isolation challenges. Martin and I are trying to get that lift from the end of Dirty Dancing. You can challenge yourself to anything. My boys have learned to ride a bike. My 2-year-old daughter is self-potty training at the moment. Any of those things is a challenge you can share, and I'm sure everyone listening at home who is suffering from this isolation is enjoying seeing those, so just add them with the hashtag, and I will give you a prize. The prize is nothing except pride, but hey, who doesn't need more pride? So all you have to do is go on Twitter. If you don't have a Twitter account, it doesn't matter. You can follow the questions and comments simply by opening it in your browser and going to the search bar. If you do want to add an account, you can even do it in a pseudonym or something where no one can know who it is and just listen. And that's the way to go about it. But we do enjoy having new members of the community showing up there. What I like is anybody at any level from anywhere can ask the speakers a question, and the speakers are enjoying getting back to you. I know Dirk is going to be busy after his talk today. It's dinner time over there right now. Maybe some of his postdocs or grad students can answer some of those questions, and he'll be able to get back to you in the coming days. So if you listen to a talk and you like it and you have a comment or a thought, put a question up there. The speakers will get back to you, and it's been creating some really great discussions that I've been enjoying looking at. I know with Sophie Treffelet's work, it actually led to discussions about techniques and approaches, and that's what I see as being a nice resource open to everyone thinking about, well, if I want to do this, how should I do it? Here are some answers I can use. All right, so I won't say any more about that. None of this would have been possible without the rapid response of these people right up here. We had 4 full weeks of talks organized within 1 week of hearing that everything was going to get shut down. And that's thanks to these wonderful speakers and their availability and their willingness to share unpublished data and work and have it put out there, not only in a live forum, but also in a recording setting, so that those of you who have other things going on at home who can't stop and listen until the kids are in bed or for other reasons can actually still enjoy these talks. And so I thank all of them for the openness and willingness to be involved and to adopt a new model, which really hasn't been the way we've been doing science. I know there are still conferences that are saying no photos, and here we are, these people are letting you record their work and watch it anytime you like. And I really congratulate them for having that foresight. And then we had an additional 2 weeks of speakers who readily accepted the challenge to join the group. And I know that the content this week has been amazing and it's going to get even better next week. And so enjoy. Tomorrow, we're going to hear from Serine Riera, who has been supported by the American Diabetes Association like myself. And so if you'd like to thank me personally, the way you can do that is to follow this link, diabetes.org/pathway. Go and read about what's going on, what kind of research they're funding. I'm sure you'll see that it's some excellent research in all realms of diabetes. And then if you feel to do so, if you make a donation in my honor, that would really make me happy. But you don't need to do that to listen to these. We're very happy to provide them for you. And like I said, tomorrow we're going to get to hear from Serine about her work on the peripheral nervous system and how it regulates energy expenditure. That's definitely going to be exciting, and I encourage you all to tune in for that. If you can't get it live, remember there are recordings going up both on our Facebook page and on YouTube. And feel free to share that with people. Share the links, post them on your own pages. We want to have this science distributed to anyone who's interested in listening. All right, so without further ado, I am going to pass the host controls over to Dirk, so you can all enjoy this wonderful talk. There's a lot of new stuff in there, so enjoy. I will see you in the Twitter space and tomorrow. All right, so Dirk, I am handing you the host controls. Now, make host. Yes. All right, stop my share. Bye, everybody.
So hello, everybody. First, I would like to thank Philip for organizing this awesome lecture series here. And also thanks again for inviting me. It's actually a great honor to present our work here in that forum. And so let's get started. So today I would like to talk about our new findings about regulatory T-cell metabolism. And it's all about glutathione, serine, and immune tolerance. So So we have the first technical issue. Okay, so in principle, my lab is interested in immunology, and on a very superficial level, the immune system exists in 2 different states. One is the immune activated state, and one is the immune tolerogenic state. And keeping the balance between both of these states is absolutely crucial for a healthy body function. So if one or the other side of the immune system is overpronounced for a prolonged period of time, that has significant disease consequences, being it autoimmunity or fostering cancer development. So keeping this balance is key, and we are very interested in mechanisms that actually confer this immune homeostatic state. So a couple of years ago, we got very interested in a new concept in immunology at that time, that is immunometabolism, that basically investigates the interplay between immunological and metabolic processes. And here you see a selection of the different T helper cell populations. And we all know that these T helper cells are important to execute specialized immune functions. However, each of these subsets is also implicated in several diseases, like Th1 cells in type 1 diabetes, or Th17 cells as the drivers of multiple sclerosis, Th2 in allergies. And so it became more and more apparent that actually incorrect metabolic remodeling can cause aberrant immune responses and therefore contributes to this disease development. And it's now our mission to interfere with the metabolic setup of these falsified immune responses and actually to mitigate these inflammatory diseases. So when we're talking about immunometabolism, we generally have pathways in mind that give rise to energy and biosynthetic precursors. And the main fuel for T cells in the cell is actually glucose. And glucose is used in a pathway that is called glycolysis to generate ATP. And this is a very fast, but also a very inefficient process. And therefore, metabolites of glucose can be also rediverted and enter the citric acid cycle, or TCA cycle, which is the central oxidative cycle in mammalian metabolism. So T cells can not only use glucose, but they can also use fatty acids and also amino acids, for example, glutamine, and all these nutrients have in common that they somehow end up in the citric acid cycle where they are subjected to further oxidation. And in this process, we generate reductant equivalents, which are then reoxidized in the electron transport chain of the mitochondria in a process that is called oxidative phosphorylation, which is the cellular most efficient way to generate cellular ATP and energy. And in this process, we generate reactive oxygen species as a byproduct. And large parts of this talk are actually about reactive oxygen species. And when I talk about reactive oxygen species, I strictly mean metabolic ROS, which is generated in the electron transport chain during ATP generation. And in general, this channeling of electron is quite efficient, and only 0.1 to 2% of these electrons get actually lost and then transferred to oxygen to give rise to this very heterogeneous group of molecules, which we sloppily term just with one name, that is ROS. And ROS in lymphocytes for a long time has been known to be important signaling mediators, and at low concentrations they can confirm cellular activation in lymphocytes. However, when ROS levels accumulate too much in the cell, they can also induce detrimental processes like senescence or cell death. And therefore, ROS has to be tightly balanced. And we actually wanted to know how this is achieved in molecular terms and in T cells. And to cut a long story short, we actually got aware of a molecule that is called the glutamate cysteine ligase that is actually important to generate glutathione. It's actually catalyzing the rate-limiting step in glutathione synthesis. And as you might remember from your biochemistry textbooks, glutathione is the most abundant cellular antioxidant. And there was actually, when we started, there was a lot of literature out there where people threw either glutathione themselves or N-acetylcysteine on different cell lines, or maybe also primary cells, and measured different parameters. And we confirmed this data, at least partially, and got convinced that glutathione indeed might have an important function in T cells. So we decided to go one step ahead, and we generated a mouse model where we actually targeted GCLC, so the catalytic subunit of the glutamate cysteine ligase, and generated T cell-specific GCLC knockout mice to study the role of GCLC and glutathione in vitro, but also in vivo. So here's a validation of these mice. So we isolated CD4 T cells as well as CD8 T cells, stimulated them via their T cell receptor. By this, these cells get activated. And as you can see here, there is an activation-induced rise of glutathione synthesis, which was measured by mass spectrometry. However, when we looked in the GCLC deficient state, that basically doesn't happen. So deficiency for this enzyme equals deficiency for glutathione itself. So at that stage, we basically hypothesized that these cells have now skyrocketing ROS and are probably prone to death. However, none of these assumptions was true. Indeed, the CD4 or CD8 GCLC deficient T cells had increased ROS levels, but it was just moderately increased. And actually, these cells were pretty healthy. And at least in the short-term activation, they also activated quite normally when compared to controls. So at that stage, that was pretty disappointing for us. However, we were more interested in the actually in vivo situation of this molecule. So we challenged control and the T-cell specific GCLC knockout mice with a virus that is called the lymphocytic coring meningitis virus, which basically elicits very strong CD8 cytotoxic T-cell responses. And in consistency with our in vitro data, we first assessed a cellular activation of these cells. And as you can see here, and also in line with our in vitro data, activation basically didn't differ between both genotypes. However, when we then looked at the actual viral control, we found, and these are viral titers here now, when we looked at the titers in the control mice that were infected, they were under tight control. So there was a valid immune reaction against this virus. However, viral titers in the T-cell specific GCLC knockout mice were just skyrocketing, indicating that this molecule and glutathione in T-cell is absolutely crucial to mount an antiviral response. But what about autoimmune reactions? So to basically study the effect of this molecule in autoimmunity, we took advantage of one of our house models, which is called experimental autoimmune encephalomyelitis, a mouse model for the human disease multiple sclerosis. And very briefly, in this scenario, we prime T cells to an antigen that is most abundant in the central nervous system. These cells recruit other inflammatory cells and basically elicit a strong neuroinflammatory reaction, which leads to demyelination and paralysis of the affected animals. So when we induce this disease in controls, we indeed could, and this is a severity score here, basically could induce a very strong ERE disease burden. However, none of the T-cell specific GCLC knockout mice developed any clinical symptoms of this disease, indicating the importance of the glutathione cascade in T cells for the progression of this multiple sclerosis-like disease. And we were really delighted by the fact that the glutathione interfering drug dimethyl fumarate at that time had been recently approved for the treatment of multiple sclerosis patients. And we think that our study actually contributed, at least in parts, to the understanding of the mode of action of this very complex drug. So what was actually happening in a more mechanistic way? So when T cells get activated, they switch their metabolic program from a more oxidative metabolism to activation-induced glycolysis, which supports the effector functions in effector cells. And this switch is actually mediated by a transcription factor, or one of the transcription factor mediating that is a cMYC. And we found that glutathione had quite some impact on MYC expression. So in more detail, we found that the glutamate cysteine ligase-derived glutathione buffered ROS levels to a sweet spot that allowed proper mTOR activation, but also NFET activation. Both of these pathways fit into the MYC pathway, stabilized MYC expression, and then MYC was important to metabolically reprogram these cells to activation-induced glycolysis, but also increased glutamine metabolism, and that was needed to mediate inflammatory reactions in vivo. However, already at that stage, we actually, something different in the beginning. So inflammation and autoimmunity can, of course, also occur if there is an imbalance of inflammatory T cells and regulatory T cells. So in general, if regulatory T cells control the activity of inflammatory cells, and if regulatory T cells are not functioning properly, then these cells basically get out of control and can induce a spontaneous or a systemic inflammatory state in an organism. So very early already in our T-cell specific knockout mice, we realized that glutathione might have a different effect on T cells when compared to effector T cells. So when we looked at effector T cells in the glutathione deficient state, we find actually a contraction of the effector T cell compartment. However, when we looked in regulatory T cells, this basically looked similar to the control state, indicating that glutathione might regulate T regulatory cells in a different way than effector T cells. So we asked then some principal questions about Treg metabolism. So far, people think that Tregs, and we actually think this is well, rely more on oxidative metabolism when compared to effector T cells. So we basically in vitro generated effector T cells, which are called here Th0s, or in parallel, we generated induced Tregs, and we induced them with TGF-beta, and then measured the respiring capacity either by mitocheck or red, or by measuring the oxygen consumption rate. And when we did this, we actually found that regulatory T cells had a higher respiratory capacity when compared to the effector T cells. So if the mitochondria is more active, one would also expect that these cells actually produce more reactive oxygen species. And so we measured that, but the result was actually exactly the opposite. So regulatory T cells had less reactive oxygen species when compared to activated effector T cells. So that was quite counterintuitive. In principle, two possibilities can arise. Either these cells have indeed a lower mitochondrial activity, which obviously is not the case here, or they have raw scavenging system in place, which are much more efficient in detoxifying ROS. And that was the next thing we actually looked for. We actually measured glutathione levels and measured glutathione content of activated effector cells versus induced Tregs. And we found that the glutathione concentration was almost 3 times higher in Tregs when compared to these effector cells in vitro. That further substantiated our initial thoughts that glutathione might have an important role in especially regulatory T cells, and forced us to basically proceed with a genetic approach. So we crossed our mutant mice with FOXP3 CRE expressing mice. So FOXP CRE is basically targeting only regulatory T cells that represent something like 6 to 10% of the overall T cell pool. And so we achieve a very specific deletion in regulatory T cells. And of course, we wanted to study the relevance of glutathione in regulatory T cells in vivo, but also ex vivo. So here is the validation of this knockout. So when we isolated or induced Treg cells, actually GCLC mRNA expression was almost completely lost. So we achieved a very specific deletion. Also glutathione levels were down. And in consistency with the reduced glutathione levels, we found an increased reactive oxygen species concentration in these cells, which is basically shown here in this plot. And then we set out to basically characterize this mouse model a little bit more deeply. And initially, we looked at T regulatory cell homeostasis. And in consistency with the findings we had from the overall T cell specific GCLC knockout mice, we also found in this knockout mouse model that Treg homeostasis was not impaired. Actually, we found also increased numbers of Tregs in the Treg specific GCLC knockout mice. And also when we differentiated these cells from naive T cells, we basically didn't find any differences among the genotypes. However, here the assay is pretty maxed out, and when we titrate down the activation stimulus, so the anti-CD3 concentration, we even find that the mutant cells differentiate to a larger extent in regulatory T cells. So these cells are basically there, and we went ahead with the analysis and now monitored the general T cell compartment of these mice a little bit more deeply and thoroughly. So starting at week 6 to 8, we actually realized one thing. We found a decline of naive T cell populations in the CD4 compartment, but also in the CD8 compartment. And this went hand in hand with a rise of effector T cells. And this is a sign that there might be some autoimmune reactions developing in these animals. And that actually was the case. At a certain point, we realized that our Treg-specific GCLC knockout mice developed a severe autoinflammatory phenotype, and that was associated with splenomegaly, but also with significant increased lymph nodes. And this started around week 8, I would say. And this is an end-stage mouse here taken at week 12. And as you can see, it's inflamed, it has skin inflammation, ruffled fur. It pretty much looks like a kind of a scurvy phenotype mouse. So it looked like that GCLC and regulatory T cells actually regulates immune homeostasis. And so we characterized this disease a little bit more deeply, and it turned out to be a multi-organ inflammation. And when we analyzed different organs, we basically found all kinds of immune cell infiltrates in these various organs, being T cells, B cells, macrophages, basically everything we looked at. And this disease was also associated with a drastic reduced lifespan, as you can see here, very significant and very severe. So we looked at inflammatory markers in the serum of these cells, and we mainly concentrated on cytokines. And in the serum of the mice, we find systemically upregulated interferon gamma and also TNF. And especially interferon gamma is associated with multi-organ inflammation. And we wanted to know what is actually the relevance of these cytokines in mediating this spontaneous inflammatory disease. And that's why we co-deleted interferon gamma in our mutant mice by a genetic approach. And by doing this, we could significantly reduce the disease burden in these animals, indicating the importance of interferon gamma for this spontaneous autoinflammation. So we have a Treg-specific knockout mouse model where we ablate the glutathione cascade specifically in Tregs. And of course, we thought that the Tregs might be not functional, and that's why we had a deeper look at Treg functionality. And first, we did a surface characterization of these mice, and we here looked at different Treg populations, induced Tregs, but also natural Tregs. And to our surprise, we actually found that most of the markers, or actually all of the markers that are associated with Treg functionality, like CTLA4, jitter, OX40, ICOS, or CD25, were expressed to a similar level. However, what we realized is that the mutant Tregs were activated a little bit more when compared to their wild type controls. And this is what you see here on CD44, CD69, and also PD1. So we hypothesized that these cells might lose their actually stability and at a certain point start to secrete inflammatory cytokines. However, when we looked at that and actually induced cytokine or tried to induce cytokine expression by various stimulus, we were actually not successful. So the mutant Tregs actually did not express interferon gamma, they did not express IL-17, and they also did not express TNF. So these cells were actually pretty stable. So the cells look like Tregs, they are stable, but do they actually suppress? And to basically investigate that, we first had a look at a so-called in vitro suppression assay, where you measure the suppressive capacity of this T regulatory T cells. And you do this by labeling effector T cells with a dye that is called cell trace violet. Then you activate these effector T cells, and then that's what you basically see here. Then these cells basically start to proliferate. And by doing this, they dilute this dye cell trace violet out. And that's why we lose the intensity here. And each of these peaks is actually representative for a proliferative cycle. And the activity of regulatory T cells can be now measured by their ability to suppress this proliferative response. And when we titrated into this effector T cells, various concentrations of functional control Treg cells, and as you can see here, we could specifically downregulate or suppress the proliferative response of the effector T cells. However, when we did the same assay using the GCLC deficient Tregs, that worked in a much less efficient way. As there was still a remaining suppressive capacity of a GCLC deficient Tregs, but it was drastically lower when compared to control Tregs. So at least in vitro, GCLC seems to impact, or the deficiency of GCLC seems to impact the suppressive activity of regulatory T cells. But of course, we wanted to know what is the in vivo relevance of these findings. And to investigate that, we took advantage of a T cell transfer-induced colitis model, where you basically transfer effector T cells into Reg- mice. Reg- mice are devoid of B cells, but also of T cells. And if you transfer T cells into this Reg- mice, these T cells start to proliferate and induce a T cell-dependent colitis, and which you basically can measure as weight loss. And the interesting thing is that this colitis can be prevented by the co-transfer of functional T regulatory cells. So when we transferred effector T cells into Reg- mice, and this is the black line here, we could induce a colitis, which is being seen here as weight loss. And this disease could be rescued by the co-transfer of functional regulatory T cells, here seen in blue. But when we did the same experiments and co-transferred with a GCLC deficient regulatory T cells, these cells could not prevent the disease, and the mice still suffered from colitis. And at various stages of this disease, we re-isolated this mutant T regulatory cells, indicating, and we actually found them, and they were not simply just dying. It was the lack of suppressive activity in vivo which prompted, or which basically made sure that this disease is still occurring. So in vitro, but also in vivo, GCLC deficient regulatory T cells lack suppressive capacity. But what is the mechanism? And what you do these days is in principle always the same. You do an expression analysis, and we did RNA-seq of either induced regulatory T cells or natural regulatory T cells. And in both data sets, the results were the same. The most significant altered gene was actually FOXP3. And FOXP3 is the master transcription factor of regulatory T cells, and it defines their function and also their identity. And when we validated these experiments, and then we validated these experiments also on the protein level, and this is intracellular flow cytometry, measuring the expression levels, the MFI of FOXP3 here in this control versus mutant regulatory T cells. And by doing this, we actually had the same finding. Yes, the mutant T regulatory cells still expressed FOXP3, but they expressed it to a much lower degree when compared to the controls. And so we were worried a bit because our mice are suffering from a significant autoinflammatory disease. So there's constant inflammation ongoing in these cells. And we were worried that this inflammation is actually impacting our results. And that's why we actually rescued the phenotype of this regulatory T cells, of the mutant regulatory T cells, by a retroviral transduction of GCLC. So basically, we reconstituted GCLC in our mutant cells, and that is basically what is shown here. And that basically gives rise to FOXP3. So it increased FOXP3 expression, indicating that GCLC, by a direct or indirect effect, at least dependent on GCLCs, increases FOXP3 expression. Next, we were focusing then on pathways that are implicated in mediating or in regulating FOXP3 expression. And one of this pathways is actually mTOR. And here again, we suffered a first surprise, because when we looked at our regulatory T cells and compared controls versus mutants, we actually found that the GCLC-specific T regulatory cells had an increased activation of the mTOR pathway, more specifically, it's now, I show you now here phosphorylation of S6, which is a downstream mTOR target. And that was very surprising for us because that was diametrically opposed to our findings which we had from effector T cells, because in effector T cells, when we delete GCLC or inhibit the glutathione cascade, it looks exactly the opposite. So we have decreased mTOR activation. So again, we were worried whether this is indeed a GCLC specific effect or whether this is just because of the inflammatory conditions from which our mice are suffering. And again, we retrovirally reconstituted GCLC, but again, expression or re-expression of GCLC in GCLC deficient Tregs brought mTOR levels down, indicating that this is really a specific or might be a specific effect. So reconstitution of GCLC in GCLC deficient Tregs increases FOXP3, but it also lowers mTOR activation. But can it also rescue the functional deficit in this mutant cells? And therefore, we did also a functional validation of this. Again, we did an in vitro suppression assay. So here is the proliferation capacity shown of activated effector T cells. And this can be suppressed when we give wild type regulatory T cells to that have been transduced with NAMT vector control. When we do the similar experiments with the mutant regulatory T cells, they don't suppress, so there's no gain in suppressive function. But if we reconstitute them with the glutamate cysteine ligase, with GCLC, they regain their suppressive capacity back to the wild type levels, implying that we're really dealing here with a GCLC specific effect. However, we were still not entirely convinced by that, and that's why we decided to generate an additional mouse model. So again, we crossed our mutant GCLC flox mice, this time with a mouse where we can induce the expression of FOXP3 CRE. So here, the FOXP3 CRE could be induced by an external stimulus, and which is beta-tamoxifen in this case, in vivo, or in vitro, you can use the soluble compound, which is 4-hydroxy beta-tamoxifen. And by this, we have a model in hand that is devoid of any inflammation, which we can basically use to confirm the cornerstones of our study. And so we did. So we isolated naive T cells from this non-inflamed, inducible Treg-specific GCLC knockout mice, induced the deletion, and measured mTOR activation as well as FOXP3 expression. And in consistency with our straight Treg-specific knockout mouse model, the induction and vitro induction of the GCLC deletion led to an increase in mTOR activity, here indicated by a 6 phosphorylation, and in parallel led to a down regulation of FOXP3. So that was actually very great, and actually we now started to believe our own data, and we asked now the question whether the increased mTOR activity is actually associated with the suppressive function, or with the lack of the suppressive function of this mutant Treg cells. And to investigate that, we did a very easy approach in vitro, but also in vivo. We either injected in our mutant mice the mTOR inhibitor rapamycin, or cultivated the mutant cells for 24 hours with rapamycin, then washed the rapamycin away and measured the suppressive capacity of these cells again. So here are the in vitro results shown. So again, the mutant cells do not suppress the proliferative response. So we still have a shift to the left-hand side. Effector cells are still proliferating. But when we use the cells that have been incubated with rapamycin for 24 hours and then put in the suppressive assay, these cells actually regained their suppressive function. So the increased mTOR activation in our glutathione deficient Tregs is associated with the lack of their functional ability. But then we wanted to know how mTOR is actually linked with FOXP3 expression. And a couple of years ago, the Delgov lab showed that in mTOR deficient cells, you have an increased phosphorylation of SMAD3. And SMAD3 binds the FOXP3 promoter and can increase FOXP3 expression. So we had the other situation. We actually had increased mTOR activation in our mutants. And so we looked for SMAD3 phosphorylation. And when we subtracted the background here, we actually saw that SMAD3 phosphorylation was actually quite minor in our GCLC deficient T regulatory cells, which is in consistency with the data that was published a couple of years ago. And when we inhibited mTOR, we could increase SMAD3 phosphorylation both in wild type as well as in a mutant to a similar degree. And when we retrovirally reconstitute SMAD3, we could activate SMAD3, and in consistency with that, we could also restore FOXP3 expression in our GCLC deficient Tregs. And of course, we wanted to know whether this could also restore their functionality. And again, we made use of retroviral transduction model. And when we transduced anti-vector controls in GCLC deficient Tregs, they were again still not able to suppress. But when we reconstituted SMAD3 in these mutants, we could reinstate their suppressive capacity back to wild type levels. So glutathione somehow leads to an increase of mTOR activation, and this basically leads to a dephosphorylation of SMAD3, and that's why we have a lower FOXP3 expression. And when we reconstitute this pathway at the level of SMAD3, we could reinstate the function. So mTOR is a regulator of metabolism. And of course, we were asking the question whether GCLC now regulates Treg metabolism as well. And first, we had a look at glycolysis. And as you can see here, so this is a classical seahorse analysis where we measure the extracellular acidification rate, which in principle is lactate. And as you can see here, there's a higher glycolytic activity in the GCLC deficient T regulatory cells. And also these cells consume much more glucose and also secrete more lactate. So, but what about mitochondrial active respiration? What about OXPHOS? OXPHOS was basically up in a similar way as seen here in the OCR, so a classical seahorse assay. So the basal mitochondrial respiration was significantly increased. And again, this was very disturbing for us in the beginning because our findings, which we published in 2017 in Immunity, were exactly the opposite. So in effector T cells, the deficiency for GCLC basically decreases glycolysis and also decreases OXPHOS. But in regulatory T cells, there seems to be a subset-specific role of glutathione, and GCLC dampens Treg metabolism. And we wanted to know whether this dampening of the Treg metabolism is actually associated with their function. So to investigate that, we went back to our in vitro suppression assays, and we either inhibited glycolysis or oxidative phosphorylation by a suboptimal dose of specific inhibitors of this pathways. So we either use suboptimal concentrations of 2DG or oligomycin. And again, our mutant T regulatory cells are not able to suppress, as shown multiple times. However, when we suboptimal inhibit glycolysis, they gain their suppressive function. And actually the same is happening when we're using oligomycin. So also oligomycin and inhibiting OXPHOS in a suboptimal way reinstates their suppressive capacity. So glutathione and Tregs really seems to dampen metabolism, and this is absolutely crucial for the suppressive function of these cells. So how is that possible? So we actually were wondering whether we were able to find a specific metabolic signature in our mutant regulatory T cells. And so we went back to our RNA-seq data, and actually we found a very striking cluster and found that basically almost all the, not all, but most of the genes that are implicated in serine metabolism are actually upregulated in glutathione deficient regulatory T cells. And also this regulatory T cells consumed much more serine. So serine is a non-essential amino acid. So it can be either taken up from the extracellular space by specific transporters, or it can be synthesized through the activity of the phosphoglycerate dehydrogenase PHGDH. And in the beginning, we just focused on serine uptake and asked the question, what is the relevance of serine uptake for the functionality of our mutant cells? So, and to do this, we basically applied a very simple system. We simply cultivated our cells in serine deficient media and then measured mTOR activation in the straight knockout mouse model or in the inducible knockout mouse model. And in both situations, in serine deficiency lowered mTOR activation, and in parallel, it increased FOXP3 expression. So serine, due to its ability to active, so basically that proved that serine is actually quite important for mTOR activation and FOXP3 expression, at least in our knockout mouse model. And in a different approach, we limited serine entry into the cell by a specific inhibition of the so-called alanine-serine cysteine transporter system. And when we did this by a drug that is called L-phenylglycine, we could also increase FOXP3 expression. However, this is just an inhibitor and it needs some validation, and we validated these results by using a CRISPR-Cas9 mediated genomic deletion approach of this alanine-serine cysteine transporter 1, which, by the way, was upregulated in the mutant cell. So that seems to be the most significant transporter in our system. There's also an AACT2, which actually is not upregulated when we compare the expression and control in mutants, but AACT1 is upregulated. And the interesting fact of this transporter is that it's actually an NRF2 target. And that actually fits very well because if you remember, our T regulatory cells have increased ROS levels, and we also found increased NRF2 activation and an increased AACT1 expression. So when we downregulate this transporter now with a CRISPR-Cas9 approach that was quite efficient, we were also able to increase FOXP3 expression again, indicating that limiting serine uptake has a significant effect on mTOR, but also on FOXP3 expression. But is this also relevant for Treg function? And so we went back to our inhibitor, so we and to our suppression assays. So we treated our mutant cells with this inhibitor L-phenylglycine and assessed the suppressive function. And again, here control cells have a significant suppressive capacity, in the non-treated T regulatory cells lack this suppressive capacity in the mutated state. However, when we inhibit serine uptake by using L-phenylglycine, we could reinstate the suppressive capacity, indicating that serine uptake is crucial for the suppressive capacity of GCLC deficient T regulatory cells. But what about serine synthesis? So serine is synthesized from glucose, as already mentioned, and that goes through this PHGDH cascade here. And I already showed you that our GCLC deficient T regulatory cells are actually more glycolytic. And we had the hypothesis that the increased glycolysis might be needed to support serine synthesis. And to investigate that, we took advantage of an inhibitor that was described and developed by Jason's Lucasales lab. And we measured the extracellular acidification rate, as well as we did isotopic tracing experiments. And so when we measured the glycolytic activity here by ECA using the seahorse in the presence or the absence of this inhibitor, we actually found that there is increased ECA as shown before in the mutants. However, when we blocked serine synthesis, we could reduce the glycolytic activity actually of both of wild type as well as of mutant regulatory T cells, indicating that regulatory T cells also in their wild type situation use a significant amount of their glycolytic intermediates to support serine synthesis. More significantly, we also found that actually the contribution of glucose into the serine pathway was much higher in our GCLC deficient Tregs when compared to controls, indicating that also glucose is redirected into the serine synthesis pathways to probably interfere with the suppressive function of these cells. So does this also impact then FOXP3 expression? And so of course we had a look at FOXP3 expression and in both situation inducible knockouts, as well as in the straight knockout Treg specific knockout models, inhibition of serine synthesis could increase FOXP3 expression. And also this was able to reinstate the function of these cells. Again, the mutant cells do not suppress effector T cell responses to a much lower degree than controls. However, when we inhibit serine synthesis by inhibition of PHGDH, we could reinstate their suppressive capacity back to wild type levels. So there seems to be a critical cellular serine concentration that actually regulates Treg function. But serine is also a very interesting amino acid because it's also used, it can be used to actually feel the one carbon metabolic network. And this is very important to support nucleic acid synthesis and to support proliferation. And of course, we asked the question whether our GCLC mutant Tregs enhance one carbon metabolism and whether this has any functional consequences. So to do this or to investigate that, we did a metabolic tracing experiment with C13 labeled serine, and we measured the end product, or one of the end products of the one carbon metabolism, which is M1 format. And as you can see here, M1 format concentrations were significantly increased in our mutant Tregs. And when we actually blocked serine uptake, again by inhibiting AACT1 and 2 format concentration in both actually wild type and the mutant Tregs could be reduced. So as already mentioned, serine and the one carbon metabolic network are important to sustain or to foster proliferation of effector T cells, which had been published by Marsha Haggins and also by Rusty Jones. And we wanted to know whether our GCLC deficient Tregs also have a higher proliferative capacity. And we were actually quite astonished that this was actually the case. And as you can see here, GCLC deficient Tregs proliferate much stronger when we do a Ki-67 staining, as well as we measure proliferation by thymidine incorporation. And why were we again surprised? Because these findings are again, they diametrically opposed to our findings in effector T cells, because in effector T cells, GCLC deficiency basically prevents any proliferative response. And this further reinstates now our theory that there is a subset-specific role of glutathione, and Treg responds to glutathione depletion in a much different way than conventional T cells. So, for what is the, as already mentioned, the one-carbon metabolism supports proliferation, and we wanted to know what is the degree of this, what is the significance of the one-carbon metabolism in supporting this increased proliferative rate in our GCLC deficient Tregs, and that's why we inhibited the serine hydroxymethyl transferase and SHMT by specific inhibition. And by this, we prevent the entry of the carbon atoms in the one-carbon metabolic network. Again, our mutant T regulatory cells, and this is proliferation now here, have a higher proliferative capacity compared to controls. And when we inhibit SHMT, we can basically bring this down to control levels, also indicating that the contribution of the one-carbon metabolism for the proliferation of our mutant Tregs is much higher when compared to controls. So does this have any impact now on FOXP3 expression? And so we of course also measured FOXP3 expression and whether this could also rescue FOXP3 expression. However, it actually didn't. So the SHMT dependent one-carbon metabolism and proliferation seems to be segregated from the mTOR dependent downregulation of FOXP3. So there seems to be two pathways in place which are both fed by increased serine concentrations in our mutant regulatory T cells. So this is the final scheme which we came up. We propose a feedback loop between glutathione and serine that controls the suppressive capacity of regulatory T cells. So in the absence of glutathione, we have increased ROS levels, and these increased ROS levels activate NRF2, so the main response transcription factor for the antioxidative response. NRF2 then drives the expression of serine transporters, mainly AACT1. There is an increased serine influx in the cells that activates mTOR and suppresses SMAD3, and this lowers FOXP3 levels in our mutant cells, which basically results in a lack of suppressive capacity. At the same time, serine supports proliferation of regulatory T cells by feeding into the one-carbon metabolism network. And this regulatory feedback loop also might explain the finding that proliferating T regulatory T cells are actually much less suppressive when compared to quiescent regulatory T cells. So it's all about serine, and we were very excited about that. And we also asked the question, so what is the relevance of serine now in vivo? And to actually get a grip on that, we used a serine, it's actually a serine glycine deficient diet to rescue our phenotype. So specifically, after weaning, we basically diverted our cohort. Some of the mice remained on normal diet, where actually half of the cohorts got the serine deficient diet, and then we basically measured the onset of the spontaneous autoinflammatory disease I talked about previously. And mice that actually remained on the regular diet, they developed this very severe spontaneous inflammatory disease, and they actually died because of this significant autoimmunity. However, mice that received a serine glycine deficient diet, they actually survived. And also, they didn't activate T cells, and there were basically no inflammatory markers detectable in these mice. And when we isolated the regulatory T cells out of this serine glycine deficient fat, serine glycine deficient diet fat animals, Tregs looked actually quite normal. So they had normal mTOR activation, normal FOXP3 levels. So this is of course highly exciting, and we think that it's very important because if you know the disease mechanism of a given metabolic disease, you can actually think about designing on a rational basis actually a diet that counteracts this disease-causing mechanism. So, however, we were still not satisfied, and we wanted to know whether we actually can use this mechanism to increase anti-tumor immunity. And we did this in 2 different models. So we injected our mutant and control mice either with B16 melanomas or MC38 cancer carcinoma cells. And in both situations, we actually found that our mutant mice had a much stronger anti-tumor immunity, and tumor growth was significantly reduced when compared to controls. So then, of course, we wanted to know whether this is really, whether this increased anti-tumor immunity was really dependent on the lack of the suppressive capacity of this regulatory T cells, or whether other compensatory mechanisms were in place here. And that's why we rescued this phenotype in vivo. And we did this by Treg transfers. Again, our mutants mice, and these are the mice which did not receive any regulatory Treg transfers, but were injected with B16 melanoma cells, had a relatively low tumor burden. And when we injected knockout regulatory T cells, this is the line here in blue, nothing, actually not much happened. So still the tumor growth was quite minor. However, when we injected into the mutant mice functional wild-type regulatory T cells, we could increase the tumor growth, indicating that it's really that it's in our model really depending on that the increased anti-tumor immunity is really dependent on regulatory T cells and on their lack of suppressive activity. So far I just showed you mouse data, but these days you're always asked for also some relevance in human models, and so we had a look in that as well, and we isolated T cells from the peripheral blood of healthy donors, differentiated these cells to regulatory T cells, and then took advantage of a very specific GCLC inhibitor, which is BSO. So BSO brought down glutathione levels, and in parallel, it increased mTOR activation, again, phosphorylation of S6 here, and decreased FOXP3 levels. And this was also dependent on serine availability, because when we cultivated these cells in a serine deficient media, that basically brought down mTOR activation and increased FOXP3 activation. So this is pretty much in line with the data I showed you from our mouse models. And very excitingly, also in the human situation, treating these cells with BSO and decreasing glutathione basically led to a lack of suppressive capacity of these mutant cells. And BSO-treated regulatory T cells were not able to suppress effector T cell responses. So this was pretty much in line with our mouse data, and also might give our findings a translational perspective. So in the end, I would like to present you our final model, where we think that glutathione restricts serine metabolism to preserve regular T cell functions. I basically have talked about all these different aspects already. And this has been done by most of this work has been done by my PhD student, Henry Kunyavan. And if you are interested in this work, this has been just recently published as an advanced online in Cell Metabolism, and will appear in print in the May issue of Cell Metabolism. So let's come to the most important slides, and these are the thank you slides. And here we see basically my lab, and especially I would like to highlight Henry. As I already mentioned before, he is a PhD student in my lab and just handed in his PhD thesis in April. So within 4 years, he was able to basically get a story accepted in cell metabolism, which in my opinion is quite remarkable. However, everybody of my lab contributed quite significant to the studies, and it was a very collaborative way how to approach these research questions. I would like to thank my entire lab for that. We are quite a very young lab and a very international one, only 12 members, but from 8 different countries, which might be normal for North America, but it's quite remarkable for Europe. And of course, I would like to thank you, would like to thank our national, but also international collaborators at the Luxembourg Institute of Health, but also at the University of Luxembourg. Especially, I would like to highlight Johannes Meiser, with whom I share a lot of projects, and we're doing quite some interesting stuff together. I also would like to mention Carsten Hiller's lab, who contributed quite a lot to the study, especially Lisa and also Yannick, our friends at the University of Düsseldorf and Marburg and Toronto, my old friend Isaac Harris, now at the University of Rochester, of course, and our friends at Yale and University of Luxembourg. And of course, I would like to thank you for your attention. And if you have some questions, just Twitter me. I might not respond immediately, but I will respond, and thank you for listening. Okay.